Hello, my dear student. Today I am here to take a biology class. You know that stay home, stay safe, but we are here to take a unique class. You know that biotechnology is a very important subject in biological science, and it's a applied biology. So today I will take a class about recombinant DNA technology. Hope you will enjoy this class very much, and my request is that please everybody must watch this video fully, and after that if you have any comments, any question, please write down on comments box and I will give full explanation with your answer later. Thank you very much for watching this video. Okay, first of all, we'll discuss about what do you mean by recombinant DNA technology. Well, recombinant DNA molecules are DNA molecules formed by laboratory methods of genetic recombination. You can say that molecular cloning to bring together genetic materials from multiple sources, creating sequence that would not otherwise be found in the genome. The DNA sequence used in the construction of recombinant DNA molecules can originated from any species. For example, plant DNA may be joined to bacterial DNA, or human DNA may be joined with the fungal DNA. In addition, DNA sequence that do not occur anywhere in nature may be created by chemical synthesis of DNA. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure and differ only in the nucleotide sequence in the identical overall structure. Recombinant DNA molecules are sometimes called chromic DNA also. You can say that chromic DNA. There has a long history about the recombinant DNA technology. Now I will discuss about the background history of recombinant DNA. First of all, the idea of recombinant DNA was first proposed by the Peter Loban, a graduate student of Professor Dale Kaiser in the biochemistry department at Stanford University Medical School. The first publication described the successful production and replication of recombinant DNA appeared in 1972 and 1973 from Stanford and UCSF. In 1980, Paul Berg, a professor in the biochemistry department at Stanford and an author of one of the first paper, was awarded the Nobel Prize in the chemistry for his work on nucleic acid with particular regards to the recombinant DNA. You are watching the figure, Mr. Paul Berg and Werner Arber, Hamilton Smith and Daniel Nathans. They shared 1978 Nobel Prize in Physiology or Medicine from the for the discovery of restriction endonucleases, which enhanced the techniques of recombinant DNA technology. Now I will discuss the steps of recombinant DNA technology. It is a very easy way. By this you can produce a recombinant DNA technology. Recombinant DNA is widely used in biotechnology, medicine and research also. Today, recombinant protein and other products that result from the uses of DNA technology are found in the essential every Western pharmacy, physicians and veterinary office, medical testing laboratories and biological research laboratories. Molecular cloning is the laboratory process used to create recombinant DNA. It is one of two most widely used methods along with polymerase chain reaction. you can say that in short form PCR, used to direct the replication of any specific DNA sequence chosen by the experiment. There are two fundamental differences between the method. One is that molecular cloning involves replication of the DNA within a living cell, while PCR, or you can say that polymerase chain reaction, replicates DNA in the test tube free of living cell. The offer difference in that cloning involve cutting and pasting DNA sequence, while PCR amplifies by copying an existing sequence. Formation of recombinant DNA requires a cloning vector, a DNA molecules that replicates within a living cell. Vectors are generally derived from plasmid, or you can say that viruses. So most of the cases derived from plasmids, from bacteria, and represent relatively small segment of DNA uh, that contain necessary genetic signals for replication, as well as additional elements for convenience in inserting foreign DNA, identifying cell that contains recombinant DNA, and where appropriate, expressing the foreign DNA. The choice of vector from molecular cloning depend on the choice of host organisms, the size of the DNA to be cloned, and whether and how the foreign DNA is to be expressed. The DNA segment can be combined by using a variety of methods, such as you can say a restriction enzyme or ligase enzyme, ligase cloning, or Gibson assembly. Gibson assembly, you will get idea about the Gibson assembly in higher class. In standard cloning protocols, the cloning of any DNA fragment essential involves 7 steps. First of all, choice of host organisms and cloning vectors. And secondly, preparation of vector DNA. Third, preparation of DNA to be cloned. 4, recreation of recombinant DNA. Fifth, introduction of recombinant DNA into the host organisms. 6, selection of organisms containing recombinant DNA. And last one, 7, screening for cloning with desired DNA insert and biological properties. So now I want to describe the full process of recombinant DNA technology. So first of all, derived genetical part and rest of the part of plasmid DNA together, you can say that recombinant DNA. And recombinant DNA process, if you want to complete this process, you have to use two major enzymes. First of all, restriction enzyme, and secondly, plasmid ligase enzyme. Now you are watching the full recombinant DNA and the cloning process. And now I will say the steps of recombinant DNA according to your syllabus. Okay. Following from the very beginning, first of all, isolation of DNA from two sources. It may E. coli or animal cell, E. coli and animal cell, not, or you have to isolation. You can use different types of enzyme to isolate DNA from E. coli and animal cell. Cesium chloride is favorite isolating component. Secondly, cutting of DNA with restriction enzyme. So you can say that restriction enzyme or like that endonuclease enzyme. So both are right. And number next, development of recombinant DNA, adhesion of both DNA with DNA ligase. So DNA ligase, you can say that biological glue. On the other hand, in case of restriction enzyme, you can say that biological knife. And number 4, insertion of recombinant DNA plasmid in bacteria through transformation. In this case, you can say this bacteria is called transformed bacteria. And number last, cloning of bacteria. You know that by the mitosis cell division, bacteria increase their number. So this portion is called cloning of bacteria. Now we'll discuss about the uses of recombinant DNA technology. First of all, first uses is recombinant chymosin. Found in rennet, chymosin is an enzyme required to manufacture cheese. It was first genetically engineered food additive used commercially. Traditionally, processors obtain chymosin from the rennet, a preparation derived from the fourth stomach of milk-fed calves. Scientists engineered a non-pathogenic strain E. coli bacteria for large-scale laboratory production of the enzyme. Secondly, very important hormones you can produce by the process of recombinant DNA technology. Almost completely replaced insulin obtained from animal sources. You can mention example pigs and cattle. For the treatment of insulin depend on diabetes, a variety different recombinant insulin preparation are in widespread use. Recombinant insulin is synthesized by increasing the human insulin gene into E. coli or yeast, which then produce insulin for human use. Number next, recombinant human growth hormone HGH, or you can say that somatotropin hormone, administered to patient whose pituitary gland generating insufficient quantities to support normal growth and development. Before recombinant human growth hormone became available, HGH for therapeutic use was obtained from the pituitary gland of cadavers. This unsafe practice led to some patient developing some chronic disease. Recombinant HGH eliminated this problem and is now used therapeutically. It has also been misused as a performance enhancing drug by athletes and others professional places. Number next, recombinant blood clotting factor 8. It is a very important factor. A blood clotting protein that is administered to patient with forms of the bleeding disorder hemophilia who are unable to produce factor 8 in quantities sufficient to support normal blood coagulation. Before the development of recombinant factor 8, the protein was obtained by the processing large quantities of human blood from the multiple donors, which carried a very high risk for transmission of blood-borne infectious disease. For example, you can mention HIV, hepatitis, etc. And hepatitis B vaccine. Hepatitis B infection is controlled though the use of recombinant hepatitis B vaccine, which contains a form of the hepatitis B virus surface antigen that is produced in yeast cell. The development of the recombinant subunit vaccine was an important and necessary development because hepatitis B virus Unlike other the common virus such as polio virus cannot be grown in vitro, vaccine information from hepatitis B found elaborately in website. Hope so. And after next, I will discuss about the diagnosis of infection with HIV, most common disease now, human immunodeficiency virus HIV, and disease name is AIDS. You know that. Each of the three widely used method of diagnosis HIV infection has been developed using recombinant DNA. The antibody test used a recombinant HIV protein to test for the presence of antibodies that the body has produced in response of an HIV infection. The DNA test looks for the presence of HIV genetic materials using reverse transcription polymerase chain reaction. Development of the RT-PCR, the full form is reverse transcription polymerase chain reaction test, was made possible by the molecular cloning and sequence analysis of HIV genomes. HIV testing is not very well known among the medical history, or you can say that medical science. Golden rice, a recombinant variety of rice that has been engineered to express the enzyme responsible for biosynthesis. The variety of rice holds substance promises for the reducing the incidence of vitamin C deficiency in the world population. Golden rice is not currently in use, pending the resolution of regulatory and intellectual properties issues. Herbicides resistance crop, commercially varieties of important agriculture crop including soya, maize, you can say that cotton, have been developed that incorporate a recombinant gene that result in resistance to the herbicides and simplifies weed control by the use such kind of technology. These crops are in common commercial use in several countries. And last one, insect resistance crop. Bacillus is a bacterium that naturally produces a protein within insecticides properties. The bacterium has been applied to crop as an insect control for many years, and this practice has been widely adopted in the agriculture and gardening. Recently, plants have been developed that express a recombinant form of the bacterial proteins, which may effectively control some insect predators. Environmental issues associated with the use of these transgenic crops have not been fully resolved. Finally, I can mention scientists associated with the initial development of recombinant DNA method recognize that the potential existence for organisms containing recombinant DNA to have undesirable or dangerous properties. At the 1975 Asilomar Conference on the Recombinant DNA, these concerns were discussed, and voluntary moratorium on recombinant DNA research were initiated for experiments that were considered particularly risky. So, I think there are concerns about the byproduct in biopharmaceuticals production, where recombinant DNA result in specific protein products. The major byproduct, termed as the host cell protein, comes from the host expression system and poses a threat to the patient's health and the overall environment. I think like that. So if any organization want to set up a recombinant DNA technology in a particular area, at first they have to get permission from the authority. So thank you very much watching this video. Hope next time I will discuss about another topics. Thank you.